Thank you so much for that really kind introduction, Or. It's so great to be here. It's really so great to be here in front of so many people whose work has influenced me so much over the years. It's a real honor. So today I'm going to be talking about challenges and opportunities in large-scale neuroimaging studies of brain development. If you have questions throughout the talk, feel free to type them into the chat. Kiara and Or will moderate. I'm an informal person. I'm happy to discuss as we go. So I'm a psychiatrist, and as a clinician, the rationale for studying brain development is increasingly clear. The dominant paradigm in psychiatry now is that major neuropsychiatric illnesses are developmental in origin. And the early onset and multiple sources of data from both translational studies in humans, as well as animal studies and epidemiological studies, suggest these are developmental illnesses. So the overall kind of long-term goal of this program of research is to describe mental illness in terms of abnormal trajectories of brain development, ultimately allowing for early diagnostics that allow tailored treatments to help our patients live better. So today, I'm actually not going to get to any of the pathology work at all, or any of the psychopathology translational studies. I'm going to talk about kind of the prerequisites that we need to get there, in particular, 3 challenges. First, defining what is normal brain development. A second, understanding individual variation in brain development. And the third, reproducibility and open science in large-scale studies of brain development. Much of the data I'm going to be talking about today comes from the Philadelphia Neurodevelopmental Cohort, or as I'll talk about later, the PNC. So the PNC was a large-scale study at Penn led by Raquel Gur and Hakan Hakanarson at the Center for Applied Genomics at CHA. And it was a stimulus grant from the last financial crisis in 2008. And it was really required we do a big study fast as part of one of the grand opportunity grants from NIH. And it leveraged Hakan's work building up at the Center for Applied Genomics, where at that time he had over 50,000 kids. Now he has well over 100,000 kids who had been genotyped and critically agreed to be recontacted for future research. That was really important because it allowed us to, in only a 2-year period, conduct clinical and cognitive phenotyping on 9,500 kids using a clinical interview administered by over 100 assessors trained by Monica Calkins in our group and Ruben Gur's computerized neurocognitive battery. Most of those assessments were actually done at home, in fact. And then of those 9,500 kids, 1,600 of them came and did neuroimaging at Penn. And that's what I'll be focused on today. And so the 1,600 kids that were imaged at Penn were all done on one scanner. At that point, it was TIM Trio, a 3T Siemens system. And it was all done without any major hardware upgrades. So it's very kind of homogeneous, high quality data. And unlike studies like the Human Connectome Project that had a long lead-in period to optimize, we had to go very quickly. So we used basically product sequences throughout with one exception. This included T1 weighted imaging of brain structure, diffusion imaging with 64 directions of a single shell, 2 fMRI tasks, a short resting state scan, and an ASL perfusion scan that was a custom sequence designed by John Detry here at Penn. The imaging sample of the PNC spanned from ages 8 to 22, peak sampling the mid-adolescent age range, even mix in males and females, and relatively uniquely for a large-scale imaging study, it reflected the Philadelphia population with an even mix of Caucasians and African Americans, which is important because it generalizes to the community that we care about. So, using that sample, I'm going to talk about these 3 challenges. The first is normative network development. I'm here going to be especially focused on how normative network development supports executive function in youth. And why do we care about executive function, and what is it? So executive function is a very broad cognitive domain that includes many subconstructs like working memory, response inhibition, and task switching. Unlike other domains of cognition that develop relatively quickly, like memory or motor function, executive function shows protracted development throughout the adolescent period and going on into young adulthood. Critically, many sources of adolescent morbidity and mortality are due to failures of executive function. And from a clinical perspective, I care about executive function because basically every major neuropsychiatric disorder is associated with executive dysfunction. So here we're interested in understanding how functional and structural brain networks develop normally to support executive function and individual differences in youth. And when I'm talking about brain networks, this is the Martinos Center, so I'm sure you guys get this, but here we're talking about brain networks that are quantified using non-invasive MRI. The nodes in the network are derived from standard parcellations here, and then the edges in a structural brain network is derived from tractography techniques with diffusion imaging, and the functional network derives from time series correlation from fMRI. This allows us to build up functional networks and structural brain networks. And the key lens that we're going to view this data through is network modules. Network modules are sets of brain regions that are tightly connected to each other and more weakly connected to other parts of the brain. They correspond to functional subsystems and align with task fMRI and also lesion studies and translational studies in animals. And so if you take functional imaging data, like this is fMRI data from the PNC, and you throw it into an unsupervised spring embedding system like the Kamada-Kwai algorithm, you see immediately there's a ton of structure in the data, because tightly connected nodes are brought close together, weakly connected nodes are pushed far apart, and you can see immediately things like here, the visual system pops out at you. Here's the default mode, and here's the motor system. There's a lot of structure in this data. These modules are obvious. And this has been shown repeatedly again and again that these functional modules, which are also called communities or subnetworks, are extremely reproducible. This is now canonical work from Thomas Yo and my mentor, Randy Buckner, which showed using data collected here at MGH Martinos, that in a discovery sample of 500, replicational sample of 500 from the human genome superstruct project, the functional networks are basically completely identical and highly reproducible. What's important when we care about executive function is that complex cognitive operations like executive tasks require coordination of many different functional network modules. So for example, for a working memory task, which is a key domain of executive function, working memory requires that you turn on the frontoparietal system and the ventral attention system, and then deactivate non-executive systems like the default mode system and sensory motor cortex. So but here we're interested in how functional and structural brain networks mature to support these patterns of activation and the behaviors of executive function. And over time, we actually have come to learn a lot about what normative brain network maturation is, in particular for functional networks. This goes back to really important, now like widely replicated work from Damien Fair, Nico Dosenbach, Steve Peterson, Brad Schlager, that shows that these functional networks become more distinct as kids grow up. They become more segregated. So you get more connectivity within a module and less connectivity between modules. This has been widely replicated by many independent groups, including in our own data here in a 2014 paper, showing that you get more connectivity within modules and less between modules. However, it was relatively less known whether structural brain networks measured with diffusion imaging undergo a similar process. And this is what Graham Baum tackled. Graham is an awesome trainee. He was a grad student with me, and now he's a postdoc here at Harvard with Leah Somerville. And I'll be talking about two of his great papers that he did while he was still at Penn. Here, Graham quantified modular segregation using a graph statistic called the participation coefficient. And the participation coefficient basically measures the degree to which a given brain region or node in the network is integrated with other network modules. And it ranges from 0 to 1, and a low participation coefficient closer to 0 indicates increased modular segregation and less participation with other network modules. And so using a large sample from the PNC and diffusion imaging, Graham showed that just like in functional networks, structural brain networks undergo a similar process of modular segregation. So here you can see on the y-axis, the mean participation coefficient, on the x-axis, age 8 to 22, and you see this gradual but highly significant decline of the participation coefficient throughout this period, indicating increasing network segregation. And I want to caution that though this is a highly significant p-value, the effect size is relatively small, reflecting the complexity of these measures and the noise in the data. So this is averaging over every node in the network. If you look at individual nodes, you can see that this effect is largely driven by the association network, by association cortices, in particular the frontoparietal system and the default mode network. Now, in terms of its functional relevance, Graham went on to show that segregation was related to individual differences in executive function, and that the more segregated the frontoparietal system was, the better kids did on executive tasks. Furthermore, this segregation of the frontoparietal system mediated the observed improvement of executive function over the age range we studied. So if you put this together, we've seen in both functional and structural imaging data that these modules, which are present early on in youth, become increasingly more distinct and segregated as kids grow up, and in part mediate the development of executive function. However, this brings up an obvious question, which is you have these two different imaging modalities that are both undergoing a similar process, but we haven't really examined the relationship between them. So in a follow-up paper, Graham looked at how coupling between structural and functional brain networks changes during youth. And here we're defining coupling in a very, very simple way. There's a lot of cool ways to do this. We opted for an extremely simple way. And here we build up a structural network using diffusion imaging and tractography, a functional connectivity network from fMRI data. And we simply look at the pattern of connectivity at each region and correlate them. So you're basically just taking one row of this adjacency matrix between these from structural imaging and correlating it with one row of the connectivity matrix and doing experiments correlation and getting a measure of coupling that's defined that way. So if you look at the overall pattern of structure function coupling, you can see immediately that it's not randomly distributed over the cortex. There's a lot of structure there. You can see there's high coupling in sensory motor cortex and the visual system. And furthermore, this pattern aligns with other known principles of brain organization. So going back to the last section, if we look at how structure function coupling is related to the participation coefficient, whether structural participation coefficient from diffusion imaging or functional participation coefficient from the fMRI networks, you can see that higher coupling is present in nodes that are more segregated. So nodes that are more segregated have higher coupling. Similarly, if you look at outside data, like the principal gradient from sensory motor cortex to association cortex defined by Dan Margulies and Johnny Smallwood, you can see that association cortex generally has lower structure function coupling, and sensory motor cortex has higher structure and function coupling. You can even look at evolutionary expansion. If you look compared to macaque and humans, you can see that cortex that is more conserved, less expanded, has higher coupling, and cortex that's more expanded has lower coupling. So this shows that structure function coupling aligns with other patterns of brain organization that we know quite well. And the next question is, well, how does that change over development? And what's interesting is just like the average pattern of structure function coupling itself aligns with this known hierarchy of brain organization, the development aligns with this hierarchy as well. In particular, you get increased structure function coupling in association cortex as kids grow up. And so this is, if you look at this, this is on the x-axis, the principal gradient. So this is association cortex here, sensory motor cortex here. And on this x-axis, you have evolutionary expansion, so more expanded, less expanded. And then on the same y-axis, you have structure function coupling changes with age. So here you see that the regions where you have the biggest increase in structure function coupling as kids grow up is present in association cortex that is highly expanded in evolution. In particular, this is most present in the default mode cortex. So one obvious criticism of this data is I'm drawing developmental inference from a cross-sectional study. However, by the time that Graham completed this work, around 294 kids from the PNC had come back for one follow-up scan. And so we looked to see how convergent the cross-sectional results were with the longitudinal within-subject change. And you can see that the cross-sectional age effects are extremely similar to within-subject change from a mixed effects model. Furthermore, again, coming back to what the behaviors we care about and the functional relevance, we found that structure function coupling, especially in lateral prefrontal cortex, is associated with individual differences in executive function and mediates the observed change in executive function as kids grow up. So if you put this together, modular segregation of both structural and functional networks occurs during youth and mediates the development of executive function. This modular architecture may allow for specialization and reduce cross-modal interference as you're trying to activate some modules and deactivate others. Furthermore, we found that coupling between structural and functional networks both aligns with and develops according to a transmodal hierarchy that spans from sensory motor cortex to association cortex. Overall, this is remarkably consistent with an evolutionary process articulated by Randy Buckner and Fenner-Creenan, where late developing distributed association cortex become untethered from rigid developmental programming, where you get tight alignment between structure and function. This may both allow for flexibility as kids grow up, but also endow vulnerability to psychiatric problems. And it may suggest that common executive deficits that are seen in many different psychiatric disorders may reflect a failure of this process. And indeed, we've seen this in our own data. One study that I'd love to talk more about but don't have time to is work by Cedric Shio, who used multi-view machine learning techniques with item-wise clinical data from the PNC and functional network data to derive data-driven measures of psychopathology, including 4 dimensions like psychosis, mood to symptoms, disruptive symptoms like ADHD and anxiety symptoms. And what was key in all these data-driven dimensions was the key feature on the brain side was there's a loss of modular segregation across all of these data-driven dimensions, suggesting this normative process can be disrupted and lead to vulnerability to all sorts of different psychiatric disorders. So in the next section, I'm going to talk about a challenge that none of the studies I talked about in the previous section addressed, which is individual variation in functional neuroanatomy. This is work led by a really fantastic postdoc in the lab, Zaishu Kui, who's finishing up his postdoc with me and on the job market now. So one limitation of every study I showed you in the previous section was that we used a standard group atlas. So this is an atlas defined by John Power and Steve Peterson, which has 264 locations across the brain. But what this assumes is that the given functional systems, like here in red, these balls that denote nodes in the default mode system, that those functional systems line up on the cortex in a one-to-one fashion that's handled by neuroanatomy. So it assumes that the default mode system is located on a similar piece of anatomic cortex across individuals, and therefore the co-registration process through the structural image handles that correspondence. However, work from many different groups has shown this is demonstrably false. This is work from Steve Peterson and Evan Gordon at WashU, from Hesheng Liu doing amazing work here at Harvard, Thomas Yo and Ruby Kong. This group has shown something similar. Janine Beischerbosch and Mark Woolrich and the FSL group has shown this. These are independent data sets using independent methods showing the same results, really showing that this functional topography or how functional networks are laid out on the anatomic cortex varies a lot across individuals. And furthermore, this variability itself is heterogeneous, and that different systems are more variable than others. In particular, higher order association cortex responsible for higher order cognitive processes like executive function is itself the most variable. So you can see here, for example, that the most variability in how these where these networks are located on the anatomic cortex is seen in association cortex like the frontoparietal cortex and the default mode system. However, all this work was done on adults, and it's really unknown how this functional topography, and again, just to make sure we're on the same page in terms of the terms, functional topography is where these functional networks are laid out on the anatomic cortex, is how this functional topography evolves in kids and how it relates to things we care about, like executive function. So that's really what Zaishu addressed in this next paper. And to delineate functional networks on an individual specific basis, this has been a very fast moving subfield, and there's many different techniques to do this. But here we use a technique developed by Hongming Li and Yong Fan at Penn and Christos Stavitsikos' center that uses non-negative matrix factorization combined with spatial regularization to delineate individualized networks in a highly accurate fashion. This was originally developed using data from the Human Connectome project, and you can see that on a single subject basis that these individualized networks delineated through NMF align quite tightly with task-based fMRI. Here, using data, we applied this to data from the PNC, and it's important to recognize that these individualized network techniques are highly dependent on both data quality and data quantity. So we use extremely stringent inclusion criteria and required that you have 27 minutes of high quality fMRI data across all the different bold runs in the study. And the first step in this process is to derive a group level atlas. And that's important because otherwise you can have networks that are so individualized that you lose correspondence across people and you can't do data analysis. So the first step is we build up a group level atlas. You can select any dimensionality, but for this study, for correspondence with Thomas and Randy's work, we selected 17 networks and then labeled these networks that we developed according to the kind of common parlance of the 7-network solution from Thomas and Randy's work in JNeuroPhys. So we derive visual networks, somatomotor networks, dorsal attention networks, visual attention networks, frontoparietal networks, and default mode networks. And this is a group level atlas that then gets tailored to individual subjects. And you can see right off the bat, if you inspect individual data, that there's a ton of variability, and that this variability itself varies by the system you're looking at. So you can see that in general, if you look at individual data, that the motor network and the visual networks are very consistent across individuals. However, like on this slide, if you look at the frontoparietal network and ventral intention network, you can see that there's a lot of difference in how these are laid out in the cortex in terms of their functional topography. And here, it's important to note that this method is a soft parcellation, so it gives you weighting at each vertex for each network, but you can discretize this for display. So as a first step, we wanted to simply replicate the results that people have seen in adults according to all these different methods and data sets. And indeed, we found that in kids, just like in adults, the variability of functional network topography is maximal in association cortex. So just like we saw on the previous slide, you get the most variability in terms of the spatial layout of these functional networks in association cortex like the DLPFC, temporoparietal junction, and superior parietal cortex, medial elements, the default mode network. And if you line these up, you can see that in general, you have the lowest variability in visual and somatomotor networks and the highest variability in the frontoparietal networks. So what's really striking is that this pattern of variability also aligns with other properties of brain organization, including evolutionary expansion, myelination, and cerebral blood flow. And here we tested the alignment using a very conservative spin test based on spatial permutations that preserves the covariant structure of the data. And you can see that this pattern of variability is strongly related to how expanded cortex is, how inversely related to how myelinated it is, and positively related to the mean cerebral blood flow. So in general, these brain networks that are highly variable across individuals tend to be expanded in evolution, tend to be lightly myelinated, and tend to consume a lot of metabolic resources as quantified by blood flow. So as a next step, we wanted to understand how these individualized networks relate to both age and things we care about, like executive function. And this is high dimensional data, but we wanted to focus on interpretability initially. And so we derived a very, very simple measure called the total network representation. And this basically quantifies how much real estate on the anatomic cortex a given functional network takes up for an individual. And since all the analysis is done in normalized space, we're not aliasing in changes in surface area that may occur in development. And what was first quite striking was that we did not find a relationship between total network representation and age in our networks. However, we did find strong relationships between total network representation and individual differences in executive function on cognitive testing. In particular, we found this highly interpretable relationship between the total representation of executive networks and executive performance in two frontoparietal networks. So the more kind of real estate given over to these functional networks on the anatomic cortex, the better kids did. Furthermore, we went beyond this very simple measure and used ridge regression with split half cross-validation and nested parameter tuning to see how well we could predict individual differences in executive function using this high dimensional data. And you can see in both halves of the data, you get pretty good prediction of executive performance on an out-of-scanner battery, R values above 0.4. If you can see here on the inset, this is the actual prediction value, and here's the null distribution, which is centered around 0, because this is completely unseen data. However, what was kind of, just to make sure we weren't missing something, as a next step, even though when we looked at the total network representation, we didn't find relationships with age, as a next step, we tried this multivariate pattern regression to see if we could predict age from the multivariate pattern of the data. And somewhat to our surprise, given that the total network representation wasn't related to age, we actually found that we could predict age even better than we could predict executive function. So here in split half cross-validation, we had R values above 0.6 in both halves of the data, and way, way outside the permuted null distribution. And so it actually took us a long time to understand this, because we know that overall these networks aren't shrinking or expanding during development. Otherwise, we would see it in the total network representation. However, what seems to be happening is that in particular in association networks, there's refinement of the borders that doesn't change the overall size of these networks, but sharpens the borders a little bit. And so you get the biggest contribution of the multivariate model at the edges of these networks. So to summarize this, we found that association cortex topography is highly variable in children, just like we've seen in adults. And the cortex with variable topography is expanded over evolution, it's lightly myelinated, and receives disproportionate blood flow. Furthermore, we found that executive functions associated with the total network representation of frontoparietal networks, which are critical for executive function. So kids who had more of their cortex devoted to networks responsible for executive function did better at those tasks. Also, even though that total network representation wasn't related to age, we still could predict age in unseen data from the overall multivariate pattern of topography due to refinement of the edges of these networks. regardless, across both age and executive function, the most important and predictive features were these highly variable elements of association cortex. And overall, these results are consistent with what we were talking about in the last section, where the networks which are most flexible and have the kind of the least hardwired, the lowest structure function coupling, are also the most variable between kids. Again, which may allow for adaptability as kids grow up and develop, but also vulnerability to mental illness. And so these results have really spawned a whole bunch of follow-up studies, which are ongoing now, and we're working on 3 or 4 good papers that we're really excited about. The first is from Zaishu, which now is focused on the clinical applicability and using multi-view learning techniques to link functional topography to clinical symptoms. Adam Pines is looking at kind of reevaluating the between network connectivity elements of this, in particular canonical patterns of network segregation, and seeing how that changes when we use individualized networks at multiple scales. Sheila Shanmugam, MD PhD psychiatry research track resident, is looking at sex differences in functional topography, and collaborative work with Desmond Oates is using these individualized networks to target TMS response. So in the last section, I'm going to talk about the third and perhaps least glamorous but most important challenge for these large-scale studies of brain development, which is reproducible neuroscience. And if you haven't noticed that neuroscience, neuroimaging, psychology are in the midst of a reproducibility crisis, you've been living in an alternate and possibly more pleasant universe where maybe other unpleasant things aren't happening as well. But in our reality, we are in a reproducibility crisis. And increasingly, the field is focused on factors that reduce reproducibility in our research. 3 of the main ones are small sample sizes, non-reproducible procedures and data analysis and analytic flexibility, and artifact and data quality. So a lot of the work in the lab is actually focused on trying to get higher quality reproducible results in these large samples. And I could talk about the way we've tried to address this. So with regards to data quality, we've been really focused on tools that quantify and maximize data quality in processing. Regarding non-reproducible procedures, we've really put a lot of effort into developing automatic, scalable, and fully reproducible pipelines. And regarding sample size, we've really we have a lot of ongoing efforts that are focused on accruing large and diverse samples that are processed and curated in the same way to allow for a test of generalizability. Because finding something in PNC is great, but I'll believe it a lot more if I found it in another data set that's independently acquired under different conditions. And so just to start with data quality, we've, over the past 8-9 years, we've put a lot of work into understanding motion artifact and sources of, and how that can bias our estimates of network connectivity. So back in 2012, when I was a postdoc, I was deep in the weeds on this, and found at the same time that Kuna Van Dyke found with Randy Buckner and John Power found with Steve Peterson, that motion artifact is a major problem for estimates of functional connectivity. And in particular, this is a major problem for studies of brain development because you don't have to scan 1,600 kids to recognize that they won't sit still in the scanner. I can just look at my 3 kids at dinner and notice they're not going to sit still. So unless we control for motion in studies of brain development, basically what we're going to be studying is not brain development, but motion. And if we can quantify this by looking at, if you would kind of increasingly conservative ways to process motion and control for motion, studies of brain development. And if you study brain development naively without using top denoising techniques or covariating for it at the group level, you will basically find a 5-fold increase in developmental changes in connectivity, but it's really just motion. That's not to say that there isn't real changes in functional connectivity as kids grow up. There are, but they're correlated and collinear with, but since age is correlated and collinear with motion, if we don't control for motion, they will be totally swamped by artifact. So since 2012, when this artifact was first described by groups at Harvard and WashU and us also, there's been a proliferation of techniques to try to handle it. And especially early on, ongoing, but still occurring now, I'm asked to review a denoising paper for neuroimage or human brain mapping once a month. And so there's a lot of techniques, and it's caused problems for investigators because on the review process, there was confusion and people would say, why don't you use this denoising technique? Or this one works and this one doesn't. There was a lot of confusion on the field of the right way to do it. People were having to reprocess their data 5 times in the review process. It was kind of a nightmare. So Roscoe Chitich, a really fantastic data analyst in the lab who's now graduated and moved on to be a bioengineering graduate student with Russ Poldrack at Stanford, took on the task of benchmarking 14 commonly used denoising pipelines and seeing how they differentially controlled for motion artifact. The main takeaway, I'm not going to get into the details here, is that there's just crazy heterogeneity in how effective these pipelines are, even though they're all in common use. So kind of the top performing pipelines that are used by groups at Penn, many of the groups at Harvard, groups at WashU, in a large sample of data, they only have like 1% of connections that are significantly related to motion after denoising. A lot of the other commonly used techniques, 90% of the connections are significantly related to motion. So the first thing is, not all denoising is the same. Some denoising does it better than others. You should, and like kind of the main standard of care is understand after denoising how related your data is to motion. Just plot it and take a look at it and report it. That you'll get very far simply by doing that. So as part of that benchmarking work, Roscoe developed software, which we called the Extensible Connectivity Pipelines, or XCP, to execute that benchmarking paper because it was really complicated and had to implement 14 different pipelines. We originally conceived of this as a nose to tail processing pipeline that went from the earliest stages through deriving the connectivity matrices and all the performance assessments. However, at the same time, Russ Poldrack, Chris Gorgolowski, and Oscar Esteban at Stanford were developing fMRI prep. And basically, fMRI prep does the first third of the pipeline that XCP implements. It just does it way better. And in the end, I don't care whether the tools we use are mine or someone else's. I just want them to work. And fMRI prep works great. So what we did is we excised the first third of XCP and made it a tool that consumes the output of fMRI prep and implements the confound regression and creates the derived measures and additional quality assessments. And this was all done by like an incredible work by Aziz Adebimpe, a postdoc in the lab who's transitioning to a staff role. What's remarkable is XCP is extremely efficient. You can run it in 3 minutes on single band data, or on 10 minutes for like ABCD style data. It's been widely used, downloaded 6,600 times from Docker Hub, and even just in the last quarter of 2020, it's been successfully executed around 100,000 times. I want to say, though, that data quality is not just a problem for functional imaging. It's also a problem for structural networks as well. Graham showed in another paper that you see very, very similar problems to what you see in functional networks and structural networks derived from tractography. And then Adam Pines, a grad student in the lab, has shown that this varies, how bad the problem is, varies by what kind of structural brain network you're building up and which reconstruction technique you're using. And some reconstruction techniques are differentially sensitive to motion artifact and differentially sensitive to brain development. As a result, when we were approaching diffusion data, it became quite apparent to us that there really was nothing like fMRI prep and XCP for diffusion imaging data. So Matt Cieslak, a senior scientist in the lab, developed QSI prep, which he just submitted it is under review now and just went up on bioRxiv last week. And QSI prep is a BIDS app that consumes diffusion imaging data that's in BIDS format. And importantly, it auto-configures to correctly process nearly any acquisition scheme. So whether it's single shell or multi-shell, if it's Cartesian grid for DSI data, or even random sampling for compressed sensing, QSI prep can configure to handle it. And it uses diverse software algorithms and kind of takes the best in class from all these different software algorithms, which is quite difficult actually, because they each have their own write-out format, all their own orientation conventions, and QSI prep converts them to a standard interoperable format so we can use and actually compare them, which was challenging in the future too. Just like fMRI prep, since it's doing things automatically to your data, you need to be able to see what it's doing. So it creates text for all the steps that it's making in a standard boilerplate, and visual reports within the HTML report it creates. Importantly, we create, just like the functional pipelines, we create QC metrics for each stage of output. And as a result, even though we just put it up on bioRxiv last week, it's throughout the development process, it started to be adopted. It has now been downloaded 2,400 times from Docker Hub. If we just look at how QSI prep performs on different data, we have, if you look at the paper on bioRxiv, you can see we applied this to Cartesian grid data that's not a shelled scheme. But if you just look at some of the common shelled schemes like ABCD, single-cell diffusion tensor imaging from the PNC, multi-cell imaging from the healthy brain network, HCP lifespan sequence, or NADI optimized multi-cell sequence, you can see that automatically configured pipelines from QSI prep reduce blurring if you look at the full width half maximum blurring, and also increase data quality according to a generalizable measure of data quality from DSI Studio. Now, we collect multimodal data from all of our studies, and ASL was developed at Penn as a particular strength of the imaging program at Penn, in collaborations with John Dept. So we're not stopping with the QSI prep, and are also in the process of developing ASL prep in collaboration with John Dept's group, Russ Polderek's group, and others. And that will be hopefully released relatively soon. Together, all of these software have an open development process on GitHub, so we can get rapidly fixed bugs from an international user base. Every commit goes through a continuous integration testing on Circle CI, and then it's automatically pushed to Docker, so it's containerized where it's going to be deployed on virtually any computing platform, including it can be deployed within managed imaging databases like at XNet or Flywheel, which is what we use at Penn. These techniques are particularly important for as you get to larger and more heterogeneous data resources, and we're using them as part of an ongoing project with Mike Millam called the Reproducible Brain Charts Initiative, which is funded by NIMH and will ultimately include over 10,000 samples looking at the developing brain, which includes the PNC, which I've talked about today, but also Mike's studies in New York, the Healthy Brain Network, which is around 3,000 scans so far, NKI Rockland sample, the Human Connectome Development, the Brazilian high-risk cohort, and many other studies. We'll include fMRI and T1-weighted structural imaging, and employ these glass box processing techniques with containerized pipelines. And critically, it's not just imaging data. It's because these samples include enriched sampling of symptomatic kids with psychiatric illness. So we're going to harmonize the clinical measures through factor analysis as well, through collaborations with Giovanni Saloom, Tyler Moore, and others. What's important is this isn't just data for us, this is an open data resource, and throughout we're going to share all the raw data, all the process data, all the code, all the containers, so people can use it and we can improve over time. I'll stop there so we have time for questions. I really just want to thank all the people who did the work. Aziz Adambimbe, Graham Baum in particular, Matt Cieslak, Zaishu Kui, Cedric Shia, collaborators like Raquel and Ruben who led the PNC, collaborators, especially in particular Danny Bassett, who's an amazing network scientist and the best collaborator a human could ask for, Taki Shinohara in statistics, Christos Stavitsikos and Yang Fan in terms of the multivariate pattern recognition work, John Dept in terms of the ASL work. I want to thank our funding agency, in particular NIMH, and all the kids and their families who participate in the studies. Thanks.